Hi guys. Okay, so we are into chapter 12. So, if you'll remember, sorry, making sure I was on the right chapter there. Chapter 11 was taxonomy and just putting things into groups, and now we are starting with the domains. First of all, the two simple domains that we're not really going to spend much time on before we move into the big domain, Eukarya, which we'll get to, and then we can go into the different kingdoms. So here we go. So this chapter covers domain, archaea, and bacteria. And typically what you find in these two groups, or these two taxons, are prokaryotes, which we'll talk about. And then we're also going to talk about viruses in this chapter. Viruses are a weird little thing. We'll get to them and kind of talk about, why they're included and why they're not sometimes. And some of the characteristics and all those kinds of things. Okay, so with domain archaea, these are prokaryotes. Remember, prokaryotes has been a while, but prokaryotes are very simple organisms. Their organelles are not surrounded by a membrane, and that typically is a sign that they are very simple, like everything in there is just kind of jumbled together. With domain archaea, remember, they are organisms that live in very extreme environments. And in those environments, they are the decomposers. So if something dies in that environment, they break it down. I mean, they eat it basically, but they break it down so that it can decompose and then be kind of go away basically. So as far as kind of things to note with these particular prokaryotes, they're similar to certain eukaryotes, which remember we are a eukaryote, in that some of their cell processes are a little more similar, replicating DNA, producing energy, cellular respiration, all that kind of stuff. Don't get too bogged down on that little point. But domain archaea, as we've mentioned, are found in very extreme environments. Hot environments, acidic, volcanoes, high salt content, all those kinds of things. They seem to thrive in areas where nothing else will live. Structurally, they have cell walls that do not contain peptidoglycan. That's just a protein in the wall that helps protect the cell. It tells other organisms' immune systems to try and get rid of that archaea in this case, and so it's kind of just a marker that is pretty easily detected. The cell walls do contain a protein called isoprene, and it helps make the cells, it gives them the ability to live in these extreme environments. Transcription and translation, which are cell processes, making RNA and then making proteins from that, are different from bacteria. That gives them this genetic difference. It's one of the reasons that they were put into their own domain. Okay, so a couple, actually 4 archaeotypes that I want you to know. These will probably be on your vocab list, but thermophiles are organisms that live in very hot environments. The temperature basically has to be above boiling for them to survive. So they live in hot springs areas. They live in like the deep thermal vents, at the bottom of the ocean. Methanogens are probably the largest group of archaea. They're in places like swamps where they're kind of buried down under the muck and the mud and the water. They don't have any access to oxygen. They are anaerobic. They also live in the intestines of animals and humans, us, and they are called methanogens because they produce methane, which is a gas, and so that can be or that is what part of their process. They're helping us break down food in our intestines, but they do produce gas. Halophiles are found in water where there's lots of salt, like the halogens on the periodic table. These are salt based or salt loving organisms. So they live in places where there's more salt content or percentage than there is in the ocean. So the Dead Sea, the Great Salt Lake in Utah is where you'll find these halophiles. And then you have acidophiles, like the name says, they are living in acidic conditions. So they live in hot acidic conditions, like in Yellowstone in the springs. A lot of times they are the ones that give color to the different rock formations around the springs. So yeah, so these are the extremes. Okay, now domain bacteria. This is more prokaryotes, so again, very simple. They their job is to be decomposers, but in kind of normal environments, normal temperatures, normal, salt content, all those kinds of things. They break down dead materials in nature. They're the reason that something that dies eventually, goes away. That's the reason, like if an animal is, dead out in the pasture somewhere, a cow dies out in the middle of nowhere, scavengers will carry it off, parts of it and all that kind of stuff. But these guys are the ones who break it down to the, so it gets kind of mixed back in with the soil, or those different things. These are very important in our environment. They also, again, are in your intestine, just like some of the archaea bacteria. They break down cells, proteins, fats. You are essentially covered in bacteria, and they're constantly working to keep some of those things off of you. Sounds gross, but we're glad they're there. Their cell walls do contain peptidoglycan, one of the proteins. And this group is divided in different ways. One of the main ones is by gram staining, and this is a staining technique, and it is working with the peptidoglycan in the cell walls. And you have gram positive or gram negative, and because of the dye that they use, gram positive bacteria have a purple color when stained because of the high amounts of peptidoglycan that they contain. Gram negative have a pink color when they are stained because of lower amounts. And so it's kind of relative, whether you're looking at pink or purple, but they are labeled positive or negative based on this gram stain. The bacteria can also be separated out by their shape. They can have a coccus shape, which is just a sphere, a spirillum, which is a spiral, and bacillus is kind of a rod shape, or like a capsule shaped. And then when they are then further identified, a lot of times they are further identified by how they're grouped. And so if you have two bacteria that are together, it has the prefix diplo. If you have just a group, it's staphylo. If you have a strepto, you have it like a string, like just a chain. And so like when you talk about like getting strep throat, what they do when they check for strep throat is they go and look for, they swab the back of your throat. Somebody leaves and actually makes a slide, and they look under a microscope, and if they see a long chain of streptococcus, then you're positive for strep. So they actually go and look and see like that purple chain on that picture there. Okay, so now bacterial structures. These are pretty similar from way back when we did these in the first semester, but you've got the cell wall. Bacteria have peptidoglycan. Then outside of the cell wall is a structure called a capsule, and this provides kind of just an extra layer of protection. It helps the cell to not dry out. Typically, the capsule is what makes bacteria hard for like your immune system to destroy. So if you get a bacterial infection, a lot of times it can survive because your immune system can't get through the capsule, which is why you take antibiotics, and antibiotics will weaken the cell wall, weaken the capsule, kind of mess up what the cell is doing metabolically, like energy it's getting, stuff it's breaking down. So then your immune system can get in and destroy it. Let's see, bacteria also have anucleoid, sorry. That's that purple glob in that picture. It's the genetic material. And again, very, very simple. It's not enclosed nicely like it is in a nucleus in other cells, but it is still kind of the blueprints of the cell. And then the flagellum, which is, of course, that long tail that helps it move. Okay, so now reproduction and genetic transfer in bacteria. Most very simply go through what they call asexual reproduction, and it's basically just mitosis. And so that picture over there with the yellow, that top one, Notice it copies the black kind of squiggle is the DNA, copies it, the cell kind of elongates, gets bigger, and then it splits in two. Typically, this is called binary fission. Binary because two things are formed, fission because it's splitting. Now, sometimes bacteria do go through genetic changes and have genetic transfers. It is not sexual reproduction. It is not a combination from two other organisms combining to make one. It is just a change in the genetic material of one cell or one bacterium. Okay, so first one is transformation. Transformation is when a bacteria cell takes in floating DNA that's in its environment and just incorporates it into its own DNA. So remember, bacteria in water, there are other bacteria in the water. If one bacteria dies, it gets broken down, and a lot of times its DNA is just kind of floating out there. Another bacterium will come along and engulf it, or engulf what's left, typically that genetic information. So in that picture, that little red squiggle is the free floating DNA. So it takes it in. Notice it can do a couple of different things. It can incorporate the red into the blue squiggle, so now it's all one kind of set of DNA, or it can form a little circle that's called a plasmid that the cell can then use to make proteins or do whatever those instruct it to do. Okay, so now transduction. This is the transfer of genetic information through a bacteriophage. That's a virus that infects the bacteria. So with this one, that virus, that bacteriophage, attaches to a bacteria cell. So that's what's happening. Excuse me, that's what's happening in this first little picture. Here's the bacteria. Oh, I thought I was writing. Here is the bacteriophage. It attaches to a cell. And then it injects its own genes into that bacteria cell. So now in the second picture, you see the green and the kind of purple squiggles right there. And the genes, the purple genes in this case, tell the bacteria ribosomes to make more bacteriophages. So it essentially just makes copies of itself. Eventually it gets so full of these copies, the bacterium cell does, it bursts open, releases these new bacteriophages into the environment. They have some of the original cell's DNA, their own, the viral DNA. They attach to a new bacterium cell and empty their genetic material in there, and the process kind of starts again. Sometimes what happens is this genetic information is good and it's useful, and if it can get passed around enough through this process, then you have something that has new genetic material or a new combination. All right, the last way that bacterium will transfer genetic information is through conjugation. In conjugation, one or both cell, usually just one cell actually, form a plasmid. And in our picture, it's that little green circle. They call it the F factor, is what it's labeled here. That little green circle, that is a plasmid. Separates out from the rest of the DNA. And then two bacterium will form what they call a pilus, which is a little tube between them. And one bacterium will pass its plasmid to the other. And so it still has it because that has made a copy of its original. And now the second cell has a copy of that. And so they have, the second cell now has some new genetic material. Okay, viruses. So this is the second part of the chapter. By the way, we'll do a little bit more in class on bacteria, like using the book and looking at uses and kind of the goods and bads of bacteria. So kind of keep that in mind. Okay, then viruses. Okay, so here's the deal with viruses. They have a very simple structure, but essentially all they are doing is carrying genetic information. They can carry either DNA or RNA. They never have both at the same time. And they have a capsid that surrounds that nucleic acid, either DNA or RNA. And then not always, but sometimes there is an envelope, and it is a lipid bilayer, so a fat bilayer, surrounds the capsid. It has proteins on it. It can be made from the cell or the organism that is infected. And it just is kind of an extra layer of protection, but not all viruses have it. Okay, so the deal with viruses is they're not cells. They don't have anything that can keep them alive. They have to infect other cells, which are typically called host cells, in order to continue on carrying on their activities. This page just has pictures of some different viruses, different shapes. They can be very geometric, with different, like hexagon shapes and attachment points, like that bacteriophage in that first picture, this guy right here. The tobacco mosaic virus, the second one that looks kind of like a stack of tires with a spring in it. That's probably the most well-studied virus in the world. Then you have the HIV virus is very circular with an envelope. The one over here, the red and yellow and blue one, that is the coronavirus, which we're all way too familiar with at this point. That's a very common family of viruses, and it gets that name because the ring around the sun is called the corona. And so if you look at it, that second picture in particular, kind of a side or a cross section of it kind of looks like the sun, and so that's where that name comes from. The one right below it, the purple circle, that's a flu virus. And then the one over the green kind of squiggly, that is the Ebola virus. And so when you're infected with these viruses and you have a good enough microscope, they can check and look and see. And they can see them if they're outside of a cell. And they are grouped sometimes by shape, a lot of times by symptoms. But these are kind of some common ones. Okay, so now some little kind of special things that are classified as viruses: viroids and prions. Viroids are infectious particles that contain a little bit of RNA. They have no capsid, no envelope. They're just genetic material in an environment. They have to almost immediately affect a host cell. They don't survive just laying out on the ground somewhere. And viroids are only dangerous to plants. People and animals are not affected by viroids. Now, prions, these are infectious particles made of abnormal proteins. There's no genetic material whatsoever in them. They're very small, and they tend to infect the brains of animals and people. And what they do is they get into a cell and they act as a template, so like a way to make a copy, I guess you would say, for brain proteins, but they're not the right brain proteins. So they get into the brain and they make the wrong proteins, and they can cause different diseases in people: the Creutzfeldt-Jakob disease in people, bovine spongiform encephalopathy, that's mad cow disease, and it causes basically your systems to shut down. And so again, prions are not super common, nor are they, I believe, that well studied, but they are considered like in the virus family. Okay, so with these viruses, there is really no ability for viruses to copy themselves. They cannot replicate their DNA. They can't transcribe that DNA into RNA. They cannot translate an RNA into a protein. They don't have organelles to do this. Remember, all they have is a little bit of genetic material. And so their main purpose, their main activity is entering a host cell and basically hijacking the host organelles and forcing that host cell to make more virus. There are two cycles that viruses follow: the lytic cycle and the lysogenic cycle. The lytic cycle is immediate. When the virus enters the cell, it causes the cell to start to produce more viruses. The lysogenic cycle is a little it's a little more slow moving, and it kind of bides its time inside a host cell until the time is right. Okay, so with the lytic cycle, this picture I think hopefully will make it pretty easy for you. So first off, the virus enters the body, and we'll talk more about how they are transferred. Some are airborne, some are through contact, some are through food, whatever it is. They are in a system and then they attach to a cell. So that little kind of orange guy right right here, he is going to attach to that cell. Once he is attached, he breaks through the outside of the cell and injects its genetic material into that cell. And so that's where this one's labeled entry. Then the viral information, the genetic information that it contains, incorporates itself into the cell's genetic material and basically takes over so that more viruses are produced. So during this little replication phase over here, it's making parts. And the cell is then forced to assemble the different parts into viruses and form these new viruses. Eventually, the cell is so full of these viruses that it bursts open. That's in lyse. That's where lytic cycle comes from, that word. And then it happens again. Those viruses go on to find new host cells, and they repeat the process. It's one of the reasons, then eventually they'll run out of host cells, or the body, the organism, whatever it is, its immune system will catch on and start destroying cells that contain the virus. But it's one of those reasons that like viruses can just be 24 hours. Like they can be, you can get over them pretty quick. It's also why a lot of times if you go to the doctor with a virus, they'll say, we just have to let it run its course, because viruses will eventually kill off all of the host cells that are available to it. So that's the lytic cycle. And then the lysogenic cycle starts out the same, and then you have kind of a waiting period, and then we get back to the lytic cycle. So the virus attaches, injects its genetic material into the cell. The virus's DNA or RNA, incorporates itself into the host's genetic material. And as that cell goes through mitosis, everything gets copied. It copies the host's DNA, it copies the viral DNA. And so you go through this, and so eventually you have a lot of cells that contain this viral information. When those cells, or when that organism that is made up of those cells goes through some kind of stress, it can be another illness, something that is tiring, whatever it is, it triggers the virus to become active. And then the cell enters the lytic cycle, produces more viral particles, bursts open, releases new. So it goes through a waiting period, but then basically gets back into the lytic cycle. Okay, and just to note, retroviruses. These are viruses that force a cell to take viral RNA and basically reverse transcribe it back into DNA. And so those, again, harder possibly for your cells to fight against, but very common. Okay, so that is that on the viruses. We'll do some more stuff in class. We'll talk about some different diseases and disorders, and then go from there. Thanks, guys.